Good morning, everyone. I hope you had a great evening and you were able to consolidate part of everything that happened yesterday. It was a lot. Well, there's going to be a lot more today. So good luck with that. Yeah, we're still waiting. So we were supposed to have a guest of honor this morning, but we didn't find a guest of honor. So that's why we're taking it slow. We still have 5 minutes on the program. So yeah, we're kickstarting a new session. So our first speaker of the morning in the so-called educational lecture series, with which we're opening every day. I have to say this is pretty much like a research presentation, but it does cover on themes that are kind of foundational in cognitive and computational neuroscience, in that we're going to be hearing a lot about brain encoding techniques. So the speaker is Alessandro Gifford. Alessandro is currently a PhD student at the Freie Universität of Berlin. Excuse me for my terrible German accent. He's working in the neural dynamics of visual cognition lab, so he's a vision scientist. His research investigates the nature of intelligence, combining data and machine learning to develop new models and theoretical insights into cognitive processes. Gifford holds a bachelor in philosophy from the University of Trento, Italy, and a master in cognitive neuroscience, and his work spans computational and theoretical approaches to understanding human and artificial intelligence. So I came to meet Alessandro recently because he's one of the organizers of the Algonauts competition. It's a machine learning competition, pitting kind of brain models against each other in terms of how well they can explain brain data. I believe this competition has had a pretty deep impact already on the field. So he's been involved a lot in kind of evaluating models, but he's also collected a lot of extensive data that are used for that kind of benchmark. And he's now proposing a new toolbox to kind of systematize and making it easier to work with those models for cognitive neuroscientists. So I think he has this, even though he's still at an early stage of his career, he has a beautiful, broad range of knowledge and experience with all the key steps of making this type of work. So welcome, join me in welcoming Alessandro.
Hey, cool. Thanks for the introduction, Dune. And also thanks for inviting to the conference. Okay. Is the volume okay? Yes. So in this talk, I will focus on a research paradigm, which is based on experimentation on in silico neural responses generated by encoding models, and argue how this paradigm can benefit neuroscientific research. By encoding models, I mean algorithms that take a specific stimulus as input, such as an image, and predict the corresponding neural response. And these predicted neural responses are going to be referred to in this talk as in silico neural responses, that is, computer-generated responses, which is in opposition to in vivo responses, namely responses recorded from real brains, for example, during experiments. And although I'll be focusing on vision, this paradigm can also extend to other modalities, such as language or auditory. I will then begin with introducing the concept of experimentation on in silico neural responses and propose a toolbox, a resource that we developed in order to promote this paradigm called the Neural Encoding Dataset. I will then give an example of this paradigm applied in a research experiment where we investigated the representational relationships between visual areas using a new method called relational neural control. And finally, I will address some of the limitations of the paradigm of experimentation on in silico neural responses and argue how these limitations can be addressed by challenges in neuroscience, for example, the Algonauts Project Challenge. So let's begin with experimentation on in silico neural responses by first talking about large-scale visual neural datasets. So large-scale visual neural datasets are datasets of neural responses to visual stimulation, which are larger than usual in the image conditions which are presented or the amount of trials. So there are datasets which extensively sample recordings in single subjects Therefore, subjects usually undergo several recording sessions. And examples of such datasets are natural scenes dataset, so fMRI responses to 73,000 images, or the Neuromod initiative, initiated by Pierre Loon here at the University of Montreal, consisting of fMRI responses to a diverse range of visual stimulation from images to movies, but also to video games, or Bold Moments, consisting of fMRI responses to over 1,000 3-second videos, or also the Things initiative, which consists of different typologies of neural data, from fMRI to EEG or also EEG, all consisting of neural responses to the same set of 26,000 images. So during the last years, we've seen an increase in both the collection and also the use of such dataset. The reasons of this is, first of all, they're multi-purpose tools, meaning that they can be used to address several different types of hypotheses, but also to perform different types of research, from hypothesis testing to result validation, also to exploratory research. Second reason is that they consist of a lot of neural data, which is well suited for the development and building and training of machine learning and deep learning model of neural data, which are well known for being data hungry. They then typically consist of naturalistic stimulation, thus promoting the understanding of how the brain works in naturalistic settings. And they're also freely available, meaning that anyone can easily access them, download them, and use them. Now, despite these many advantages, however, large-scale neural data sets also come with limitations. Specifically, they're expensive to create, they take a really long time to acquire, and once acquired, they're restricted to the stimuli that were presented during data collection. So now the question is, how can we retain the benefits of these large-scale neural data sets while at the same time addressing these limitations? And one way we can do this is by using in silico data generated by encoding models. Because encoding models, this in silico data from encoding model is cheap and quick to generate and is unrestricted to any stimulus. Meaning that once an encoding model is trained, it can be deployed to generate in silico neural responses to any type of stimulus. And specifically what we can do, we can leverage large-scale neural data sets to train encoding models, and then use these encoding models to generate in silico neural responses to images or to other types of stimulation, and finally perform experiments on these in silico neural responses, where these experiments can take the different types that we're all used to, from hypothesis testing to method development, exploratory research, results validation, model development, and also neuro AI research. So then, the advantage of in silico neural responses from encoding models is that they can easily scale up to neural responses to hundreds of thousands or even millions of stimuli. And this scaling up, this in silico neural response generation is resource efficient in that it saves enormous amount of time and money, and as a consequence can greatly speed up research. Just imagine, instead of taking you weeks to collect some neural responses, you can have an encoding model generating the same amount of data in only a few minutes, and as a consequence you can test and try out way more things. Now, this does not suggest that traditional data collection efforts should be eliminated. Instead, this framework proposes a virtuous cycle where fast-paced research is performed on in silico data, its findings are then validated on in vivo data, and the novel understanding coming from this validation in turn informs new fast-paced in silico experimentation. So then, to promote this research paradigm, we built encoding models, we trained encoding models on neural responses to visual stimulation. And since we wanted to provide users, provide the community with encoding models with both a good spatial and temporal resolution, we trained encoding models on both fMRI and EEG responses to naturalistic images. For fMRI, we trained feature-weighted receptive field encoding models using the natural scenes dataset. fMRI responses from 8 subjects to 73,000 images. And as you can see from the encoding accuracy results, the models had higher prediction accuracy for early visual cortex, where they accounted for up to 65% of the explainable variance for V1, and accounted for up to 40% of the explainable variance across the ventral stream. For EEG, we trained vision transformer-based encoding models using Things EEG2, data set of 10 subjects with responses to over 16,000 images from the Things database, where these images consisted of objects on naturalistic background. And also here, their prediction accuracy reached the noise ceiling level at around 100 milliseconds, where the noise ceiling is indicated by the gray area. And the prediction accuracy remained close to noise ceiling level across the entire epoch. So then what we did is that we made these fMRI and EEG encoding modules public in an open resource called the Neural Encoding Dataset. And created a GitHub package so that you can use these trained encoding models to generate fMRI and EEG responses to any image of your choice with one line of code. To help you familiarize with the resource, Neural Encoding Dataset also comes with tutorials and data manual. And to facilitate research, Neural Encoding Dataset also comes with pre-generated fMRI and EEG responses to 150,000 images from 3 of the most widely used image sets in neuroscientific research. And finally, Neural Encoding Dataset is designed to be scalable and can rapidly expand. For example, to new data types, such as ECOG data or animal data or other data sets, neuroimaging data sets, but also to other modalities. Now, encoding models are trained on visual responses to images, but it could also be trained to neural responses to linguistic stimuli or auditory stimuli or videos instead of still images. Or it could also expand to better encoding models that better predict neural responses to stimulation. And to make this possible, we strongly welcome ideas and collaboration. So then to summarize this first part, I talked about in vivo large-scale neural data sets, talked about their benefits and how they're widely used in the community, but also addressed some of their limitations, specifically that are expensive to collect, slow to acquire, and limited to the data presented to the stimuli presented during data collection. And then proposed in silico neural data sets as a way to address these limitations, in that in silico data is cheap and quick to generate and is unrestricted to any stimulus. I then talked about the paradigm of experimentation on in silico neural responses generated by encoding models. Talked about how this could produce a synergy between in silico data and in vivo data, and also introduced the neural encoding data set, namely a resource consisting of trained encoding models accompanied by a toolbox that you can use to generate in silico neural responses to images and use them for experimental purposes. So now I will move to an example of this paradigm of experimentation on in silico neural responses in action. And specifically, I will give an example of how we use this paradigm to investigate the representational relationships of visual areas using a new method called relational neural control. The topic of this project is representational content, that is visual information encoded in neural responses. We know that human vision is mediated by a complex interconnected network of multiple brain areas that jointly process visual information, where each of these areas informs idiosyncratic representations of visual stimuli, meaning that each of these areas represents visual stimulation in a different way. Over the last century, neuroscientists have tried to understand the representational content of each area. And, for example, seminal work in this direction successfully characterized the role of early visual stimuli, visual areas, for the representation of oriented edges, or of higher visual cortical areas for representing more complex visual stimulations, such as faces. But despite these advantages, we still do not fully understand the representational content of the visual brain. So here I will talk about 2 steps that we made in order to better understand representational content of the visual cortical network. Where the first step addresses the fact that usually different areas, say V1 and FFA, are studied in isolation of each other. But studying areas in isolation comes at the cost of an incomplete characterization of the visual system as an interconnected network. So when it comes to investigating representational content, we shifted our focus from asking what does each area represent to asking, what are the representational relationships between areas? In other words, what representational content is shared between areas? So what are the visual properties, properties of visual stimulation that are similarly represented across different areas? Or what representational content is unique to a specific area? So what are the properties of visual stimuli that are differently represented between areas? So this was the first step, from studying areas in isolation approach to studying their representational relationship. Whereas the second step addresses the fact that representational content is usually studied using limited experimental picked stimuli. But using small stimuli set might risk to reproduce experimental biases, while at the same time missing important neural signals that would be available from a broader sampling. So when it comes to investigating representational content, we used a large sample size, so to evaluate a larger, broader, more diverse, and thus less biased hypothesis space. And to study representational content when following both of these steps, we developed a neural control method called relational neural control. Addressing the first step, to investigate representational relationships between areas, for example, V1 and FFA, RNC searches for images, for controlling images that either align the responses of these two areas, or disentangles the responses of these two areas, under the assumption that alignment will highlight shared representational content, and disentanglement will highlight unique representational content. And so in this case, aligning images would be images that two areas represent similarly, and disentangling images would be images that two areas represent differently. And the goal of RNC is to find, across the entire hypothesis space, across all of the images that satisfy these criteria. And as for the second step, larger hypothesis space, in order to find these controlling images, RNC searches across fMRI responses to thousands of images generated thanks to the neural encoding dataset. So it searches neural responses generated by encoding models. It works on in silico data. So here I will then show you how we use RNC to investigate representational relationships for early and mid-level visual areas in humans, V1 to V4. And specifically, we use RNC to investigate representational relationships between pairs of areas. So V1 to V2, V1 to V3, V1 to V4, etc. And I will be referring to relational neural control with RNC, which is the acronym. Okay, so the first step, we began by generating in silico fMRI responses for a large set of visual stimuli. Specifically, we used the trained encoding models from the neural encoding dataset to generate fMRI responses for all of the 8 subjects on which encoding models were trained, because NSD has 8 subjects. And again, these models were trained on the natural scenes dataset. And so we obtain in silico fMRI responses for 73 images for all of these subjects. And these are the data on which we then applied relational neural control. Now, some of you might be asking, why didn't you use NSD in the first place? Because it's already a large data set, it's responses from real brains, so there's no model in between. Why not applying relational neural control directly on that data? Well, and there are 3 reasons for this. The first reason is that within NSD, each subject only saw up to 10,000 images, whereas we generated 73 fMRI responses for 73,000 images for each subject, meaning that we increased the hypothesis space of each subject by a factor of 7, and in doing so, likely reduce experimental bias. The second reason is that in NSD, only 1,000 images are shared across subjects, meaning there are only 1,000 images that all subjects saw. And this is problematic when you wish to cross-validate your results across subjects, because you would only be able to cross-validate results for this set of 1,000 images. Instead, with the in silico data that we generated, the possibility to cross-validate extends to all 73,000 images, therefore promoting a more strict cross-validation. And finally, since neural noise is not predicted from the visual stimuli, Encoding models tend to model the signal and not the noise in the data. And as a result, the predicted neural responses, the in silico neural responses, are a denoised version of actual brain responses, which also reduces the effect of noise in the results of the analyses performed. So then together, the neural encoding dataset provided accurate and denoised in silico fMRI responses for thousands of images, which was the basis that we used to investigate representational relationships. And we started by using relational neural control to uncover the representational relationships between univariate fMRI responses of in silico data. Where by univariate responses, I mean the voxel average response across all voxels within each area. So the univariate response for V1, for example, would be the average response across all voxels which constitute V1. And to achieve this, relational neural control searched the full hypothesis space, consisting of the entire 73,000 NSD stimuli, for images that would either align the univariate responses between two areas, thus highlighting shared representational content, or disentangled the univariate responses between two areas, thus highlighting unique representational content. And here you have a plot of univariate responses, graph univariate responses. On the x-axis, you have univariate response of one area. On the y-axis, univariate response of another area. So here, alignment consisted of image conditions which drove the responses of both areas or suppressed the responses of both areas. This is what we classified as alignment, whereas disentanglement consisted of controlling images that would drive the response of one area while suppressing the other, and vice versa. And importantly, we cross-validated the results, meaning that we looked for these controlling images using 7 out of 8 subjects, the neural responses for 7 out of 8 subjects, and then testing them on the left-out subject until every subject was left out once. So all of the results that I will be showing you from now on are cross-validated. And I will start with the V1 versus V4 comparison. Here on the x-axis, you have the univariate response for V1. On the y-axis, the univariate response for V2. The cloud of black dots consists of the univariate responses for all 73,000 images, so the entire hypothesis space. And the colored dots consist of the univariate responses for the controlling images from the control conditions. And as expected, you see that the control condition, which were supposed to drive the responses of both areas, here denoted by the green dots, falls on the top right corner of the cloud. The control condition that was supposed to suppress the responses of both areas, here shown in light blue, falls on the bottom left of the manifold cloud. The control condition that was supposed to drive V1 while suppress V4, denoted in yellow, falls on the bottom left, bottom right, and the control condition driving V4 suppressing V1 falls on the top left. So this shows that univariate relational neural control was successful in finding images that align and disentangle the responses for univariate responses of V1 and V4. And what you can also notice is that univariate responses between these two areas are actually quite correlated. You see the correlation, positive correlation shape of the image manifold, and this correlation has a strength of, as an R of 0.65. And here are the same results, only plotted against significance. Here's baseline. Here the baseline is denoted by horizontal dashed lines and consists of univariate responses for randomly sample of images across the entire hypothesis space. And we observed similar results also for all other pairwise area comparisons. So this indicates, again, that RNC was successful in aligning and disentangling univariate responses between areas, and also that a large portion of univariate response representational content is shared between areas, as indicated by the positive correlation between the image manifolds. So we next asked whether there was a relationship between how well could two areas be aligned or disentangled, and how far away these areas are from each other. And for this, we used the stepwise area distance, where areas adjacent to each other would have a stepwise area distance of 1, for example, V1 to V2, V2 to V3, V3 to V4. Areas with another area in between would have a stepwise area distance of 2, for example, V1 to V3 or V2 to V4, and V1 to V4 would have a stepwise area distance of 3. So then we asked, how well could these areas be these areas could be driven or suppressed with respect to baseline as a function of how far they are from each other. And for this, we plotted the absolute univariate response difference from the baseline as a function of the stepwise zero distance. And what we found is that for the disentanglement condition, again indicated by the yellow and red dot, we saw that the univariate response difference from the baseline increased as a function of stepwise zero distance, indicating that the further away two areas were from each other, the more easily their univariate responses could be disentangled. And similarly, we also plotted the correlation of the image manifolds as a function of stepwise zero distance. Again, the image manifolds were the correlation consisted of by that correlation, I mean the correlation between the univariate responses for all images, so for the black cloud, between two areas. And again, we found that correlation decreased as a function of stepwise zero distance, indicating that the further away two areas were from each other, the more decorrelated their univariate responses would be, which suggests that unique representational content increases as a function of cortical distance. So we then asked, what do these controlling images actually look like? So here I will show these controlling images for the V1 versus V4 case. And these are the images that drive responses of both areas. They consist of cluttered scenes, many objects inside, and high spatial frequency content. These are the images suppressing both areas. Unsurprisingly, they are empty images, typically skies. These are images driving V1 while suppressing V4, typically consisting of high spatial frequencies, such as vegetation. And these are images driving V4 while suppressing V1, typically consisting of one or multiple objects on an empty background. And these are the images for the baseline condition, which, as expected, since these images were randomly sampled for the entire hypothesis space, do not show consistent visual patterns. So then looking at the disentanglement conditions, so this condition and this condition, what we inferred is that, from visual observation, is that V1 is driven by high spatial frequencies, and V4 is driven by object-like shapes, which is in line with what we know from previous research. And interestingly, object-like shapes, so the feature that drives V4, and spatial frequency, the feature that drives V1, are also co-occurring in the images that drive both areas, and are also both absent in the images suppressing both areas. So from this we concluded, again from visual inspection, that spatial frequency and object-like shapes are likely the visual features leading to unique representational content for the V1, V4 comparison when looking at univariate responses. So then so far, I showed you how we use univariate RNC to investigate the representational content for univariate responses, that is, voxel average response. But by averaging across voxel, we lose visual information that is contained in the population pattern, in the population, and the population response across all of these voxels, which are called typically multivariate responses. And these multivariate responses could encode complementary visual information that is lost when averaging across all voxels. So then the idea was to use RNC, relational neural control, to look at representational relationships with taking into account multivariate responses. Now the problem, however, is that multivariate responses are not as straightforward to compare. First of all, different areas have different amounts of voxels, meaning that they live on different dimensional spaces. And even if two areas had the same amount of voxels, there's not a one-to-one mapping between one voxel in one area and the voxel in the other area. So then we need to find a way to bring the multivariate responses of two areas to the same format, to the same space, such that they can be compared. And for this, we used representational similarity analysis. And here I will briefly give a hint of how representational similarity works. So let's say that we have two stimuli, hand and umbrella, and we record fMRI responses for these two stimuli from one area, let's say V1. So here you have fMRI response of V1 for hand and umbrella, and you see that these responses are different as expected. And then what we want to do is that we want to transform these responses from voxel space to a common space, another space, which then would be shared across areas. And for this, we computed the similarity between the multivariate responses for these two objects, and we insert the similarity score into a representational dissimilarity matrix. So you can see that this representational dissimilarity matrix is organized by conditions. It is symmetric, and each entry indicates how dissimilar the multivariate responses for two conditions are. And we can do the same for another area, say V2, and all of a sudden, these two areas are now comparable because we transformed their multivariate responses from voxel space to image similarity space. And then we can just correlate these two RDMs to assess the similarity of their multivariate responses. So this is what we did. We then used multivariate RNC to investigate representational relationships with respect to visual information contained on the in silico multivariate responses for areas V1 to V4 using representational similarity analysis. And once again, RNC searched the entire hypothesis space of 73,000 stimuli for images that would either align the multivariate responses of two areas, thus highlighting shared representational content, or disentangle the multivariate responses, thus highlighting unique representational content, where alignment consisted in images leading to similar RDMs or RSMs, and disentanglement consisted of a batch of images leading to different, dissimilar RSMs. And once again, the results are cross-validated across subjects. And these are the results that we got. And as we can see that the algorithm worked well in that it was successful in finding images that well aligned the multivariate responses of two areas. Here, these are indicated by the purple dots on top. And they have a correlation score close to 1, so it's close to ceiling. And RNC was also successful in finding image conditions that disentangled the multivariate responses, here indicated by orange dots, with respect to baseline. Here, the baseline is indicated by the gray dots, and again, consisted of RSMs deriving from random sampling of images across the entire hypothesis space. So RNC was successful in aligning and disentangling different areas as for the multivariate responses. And again, we asked whether there was a relationship between how well could these two areas be aligned or disentangled and their distance to each other. So what we did to assess this, we plotted the results as a function of stepwise zero distance. And we found that for the baseline and disentangling condition, there was a decrease in correlation between their RDMs or RSMs as a function of stepwise zero distance, indicating even, again here for the multivariate control case, that further away two areas were from each other, the more easily their multivariate responses could be disentangled. And therefore highlighting that unique representational content increases as a function of cortical distance. Again, we looked at what the images look like, focusing on the V1 versus V4 comparison. And these are the images aligning the responses of both areas. These are the images disentangling the responses, and these are the baseline images. And what you can notice is that the aligning images often contained uniform portions, such as the sky on the upper F. The disentangling images did not, and the baseline images did so, but to a lower extent. So to gain some more intuition of what's going on, we then looked at the representational similarity matrices for these images. And these are the RSMs for the alignment images. As expected, they look really similar between V1 and V4, because this is the alignment condition. And what you could also notice is that they consist of really strongly positive correlation scores, close to 1, or really strongly negative correlation scores, close to -1. And these are the RSMs for the disentanglement conditions. As expected, they do not show any visual consistent patterns between each other. So we then zoomed in onto these alignment images RSMs. And what we notice is that when an image containing the sky in the upper half when its multivariate responses were correlated to the multivariate responses of another image containing the sky in the upper half, this resulted in a strongly positive correlation. Whereas when it would be correlated, the multivariate responses for an image containing the sky were correlated with the multivariate responses of an image not containing the sky, this would result in a strongly negative correlation. And so we hypothesized that this has to do with the retinotopic properties of V1 and V4, specifically due to retinotopy, uniform portions of an image, for example, the sky, will lead to suppressed responses for all of the voxels which are tuned to that portion of the visual field. And in contrast, voxels tuned to the lower part of the visual field here would be driven. So for this image, half of the voxels more or less would be suppressed, half would be driven, which would be the same for this image. The same voxels would be driven or suppressed, which then would lead and explain this strongly positive correlation. Whereas in this case, when an image containing the sky, where half of its voxels are driven, half suppressed, is correlated with an image not including the sky, where all of the voxels are driven, this results in a negative correlation. So then, what we concluded for this is that V1 and V4 are aligned due to shared retinotopic properties. And finally, up to now, we showed that RNC solutions generalize across the in silico fMRI responses of different subjects. But in order to fully validate these results, we had to control whether they would also generalize to in vivo responses from real brain. So we did this for the V1 versus V4 comparison. Specifically, we took the controlling images from the univariate and multivariate control case, and we presented them to 6 new subjects, independent subjects, in a new fMRI experiment. And as expected, we found that the results generalized both for the univariate case and also for the multivariate case, which first of all validates RNC as a method in order to investigate representational relationships between areas, and also confirms the V1 and V4 representational relationships and controlling features that we found on in silico responses. So to summarize this part, RNC uncovered representational relationships, that is, unique and shared representational content, between early and mid-level visual areas using a larger set of in silico responses available in vivo, which resulted in both quantitative and qualitative finding. From the quantitative point of view, we found that a large portion of representational content was shared across areas, and that unique representational content increased as a function of cortical distance, which could be due to, for example, an increase in anatomical connectivity, a decrease in anatomical connectivity with increasing size between two areas. The further away two areas are, the less connections they have. But also to gradual changes along the ventral stream, such as increasing receptive field sizes. From the qualitative point of view, we found that spatial frequency and object-like shapes determine unique representational content for univariate responses of V1 and V4, and that topological image properties determined shared representational content for the multivariate responses of V1 and V4. Here, topological image properties, I mean, for example, empty regions on an image, such as the sky in the previous example. And this is interesting because what you can notice from here is that the visual features determining unique and shared representational content changed when controlling univariate and multivariate responses. Again, for the univariate case, it was object-like shapes and spatial frequencies, and for the multivariate case, it was topological image properties, which suggests that univariate and multivariate responses might capture complementary representational content, and in turn promotes integrated analysis of different levels of neural responses. Here we looked at the univariate response level, multivariate response level, but it could extend further, for example, looking at latent spaces of neural responses or their geometrical properties, or also frequency or oscillatory properties if you're looking with time frequency data. And then to link this back to the interplay between in silico and in vivo data, we used the in silico fMRI data from the neural encoding datasets to look for the controlling images. So to thoroughly explore the visual space to search for the controlling stimuli and then validated these controlling images on in vivo fMRI responses of new subjects, thus supporting the paradigm of experimentation on in silico neural responses followed by validation of results on in vivo responses. And specifically here I give you an example of this for method development, so development of relational neural control, and for exploratory research that we explored large hypothesis space in search for controlling stimuli. So now, in this last part of the talk, I will address some of the limitations that of the paradigm of experimentation on in silico neural responses, and propose how these limitations can be addressed by challenges, for example, the Algona's project challenge. Now, two important limitations of experimentation on in silico neural responses are, first of all, the limited prediction accuracy of the encoding models with which the in silico neural responses are generated, and also their limited ability to generalize out of this distribution on which they're trained. But the good news is that these limitations are in principle overcomable. For example, thanks to initiatives that aim at improving the state-of-the-art of encoding models of neural responses. For example, sensorium competition or brain score benchmark. Here, however, I will focus on another challenge called the Algona's project challenge, which is a challenge started by our group in 2019 and consists of a platform that aims at bringing neuroscientists and computer scientists together in order to build the best encoding models of the brain. So then, how this challenge works is that we provide participants with neural data and stimuli, and we ask them to use these data in order to build their encoding models, models that go from stimulus to neural responses. And then we test these models to assess how well they perform in predicting neural responses, and then choose winners. So far we had 3 editions. In the 2019 edition, which was the first edition, the neural data consisted of fMRI and MEG responses to 288 images. In the 2021 edition, data consisted of fMRI responses to 1,102 videos. And in last edition, 2023, the neural data consisted of fMRI responses to 73 images. And you can notice how along the different sessions, along the different editions, we're trying to increase the size of the data set on which the encoding models are trained. Because the more data these encoding models have, generally, the more accurate they will be in describing neural responses. So in this spirit of scaling up, providing more data, we are in the process of preparing the 2025 edition of the challenge in collaboration with CNeuromod, again, massive data set of fMRI responses from the group of Pierre Loon. And specifically, as model training, we will provide over 60 hours of fMRI responses per subject from the episodes, viewing of episodes from Friends. And we hope that the unprecedented size of this training data will result in encoding models which will improve the state of the art in neural prediction. And second, these models will then be tested on out-of-distribution movies. In this way, also promoting encoding models that better generalize out of distribution. So then, to summarize what I talked about during the talk, I started by talking about introducing the paradigm of experimentation on in silico neural responses, and also introduced the neural encoding dataset, which is a resource that we developed in order to promote the paradigm. I gave an example of how this paradigm can be applied to research. For example, giving an example of a research project where we investigated representational relationships between visual areas with a new method called relational neural control, and then introduced some of the limitations of this research paradigm, specifically limited prediction accuracy and out-of-distribution generalization of models, and proposed challenges as a way to address these limitations. So then, to conclude, my vision for the future of computational neuroscience is that larger neural data sets and challenges will promote the development of better encoding models of neural responses, which will generate more accurate in silico neural responses, leading to an increase of experimentation on these in silico neural responses, ultimately resulting in faster scientific development. I thank my collaborators and funding sources who are contributing to this future. And if you like to learn how to use neural encoding dataset or relational neural control with tutorials, you can come to my hands-on session this Friday as part of main educational at 1 p.m. Thanks.
Thanks so much, Alessandro, for this beautiful start of the day. I see we have a few minutes for questions, so don't hesitate. I'm going to start with just a remark. We started this meeting, like 7 years ago, and there's always been this challenge. We want to be at the interface of neuro and AI, and it's clear how the neuroscience field can benefit from AI, but it wasn't always clear how the AI field could concretely benefit from neuro, beyond just like inspiration at a high level. How many people in this room are really from an AI background? A little show of hands. That's it. It's not a lot. And this is a trend. But I have to say that I see AI students struggling with really being invaded by Google and Meta and having a hard time competing with their own research with this kind of large company. This is a great space to do that because those data sets are at a size that you can actually, handle. And Google and Meta have not yet seen that these competitions are actually a great space to be present in. So you probably have a few years to actually, have the lead on there. And also, if you want to test if your AI models are actually more brain-like and not just by hand-waving, that's a place where you can do it quantitatively in a very rigorous fashion. So I think this type of novel competition are really giving you a space as AI researcher to benefit from neuronal insights at a very quantitative, scientifically grounded manner. So we have one question. Let's go.
Okay, great talk. Thank you. So I had a question about your representational analysis in the visual system, and I was wondering sort of what next steps you're thinking of looking at with that, because it seems like there's many different directions you can go. It seems that perhaps you could go in a direction of looking at parallel versus serial processing within the visual system, maybe looking at orthogonal representations and how they might be correlated across different levels of visual cortex. I'm sort of wondering if you've been doing that or if you're going in that direction.
Yeah, so I do not currently have plans to work on this longer, but I have ideas about regarding how it could be applied to. So I think some interesting ones would be, first of all, addressing different neural response levels and controlling different neural response levels. I was showing here how controlling univariate and multivariate responses result in a different qualitative insight. So why not looking at other properties? For example, in fMRI, it could be even simple stuff, such as instead of looking at the average univariate response, look at the variance. What is that telling you? Or looking at latent spaces. This would be one. I think the method is could also be
maybe different covariances across the regions, right? That's sort of what I was thinking of.
You can optimize you can choose your own score, your own neural response space, and you can always optimize for that. You can always then compare the responses for different areas in order to align them or disentangle them. Another use case would also be using the method in order to align and disentangle not different areas from the same subject, but different areas across species, for example, across individuals or between humans and machines. It's a comparative method in the end, so you can compare whatever system you want.
Great, thanks.
Okay.
All right, I'm seeing people not caffeinated enough.
Thanks, that was a great talk. I have a question regarding the in silico data, and I want to know how, so I'm sure some people would say, well, if you want to generate data in silico, synthetic data, based on large data sets, how large, how diverse do those data sets need to be before we can claim that we can, because at some point we might be generating, we want to use that data for, as you said, for discovery. We want to use that then also then to generate new experiments. So in an ideal world, we'd have enough data that covers all the diversity and all the richness of real data. So then we can use that to generate new data sets. But if the data we have does not encompass enough of the diversity that is required, we'll be generating data that is shallow, that is not interesting enough, or that's just going to be covering the main things that we know already and doesn't go beyond that.
Yeah, that's the problem of out-of-distribution generalization. Actually, there are two problems that I see there. One is, I think the question is, I feel like we have a lot of brain data covering a lot of different visual stimulation types, from images, synthetic, naturalistic, to videos, and multimodal. The problem is that there's no straight way now, so far, to integrate all of these data sets together. Namely, encoding models are typically trained on single subjects on a specific data set. So one step forward would be to find ways in order to train encoding models using all of the data available, because then you would have access to a very large visual space that much larger than single data sets. So that is one problem. The second problem with in silico experimentation or in silico data from encoding models is that the task you perform arguably really much matters. So all of these encoding models that I showed were trained on visual responses to passive viewing. So subjects were not performing any task. So by definition, probably, if you wanted to study memory, short-term memory, attention, or any other thing which involves a task, you cannot do it with these models, most likely because they do not have them in their system. They were not trained with these types of responses. So they are so far mostly suitable for hypotheses that you can test by passive viewing. And probably this will also change in the future. It's just that I think that recording data with a task takes much more time, and as a consequence, you have much less neural data than just with passive viewing. Yeah.
Okay, thank you. Thank you very much.
Yeah.
So thanks again, Alessandro, for the beautiful talk.
Oh.